to the far left here, what I have is the SARS-CoV-2 virus, and this is the virus that is causing COVID-19. And on the surface of this virus, what there are things referred to as spike proteins. And so these spike proteins are things that we currently believe are involved in how this virus is able to bind onto host cells and how it's able to infect those host cells. According to the central dogma of biology, the DNA that exists in all of our cells inside of our nucleus is transcribed into RNA, which is then translated into proteins. And so the spike protein came from RNA. And so if we go a step backwards here, there is some kind of RNA sequence. There is some kind of ticker tape sequence of AUGC that codes for the spike protein that is on the surface of this coronavirus. After the researchers in China were able to sequence the SARS-CoV-2's genome, is able to identify the RNA sequence that coded for the spike protein on this virus. And so that was very important because what this now means is that we're able to synthesize these proteins as well as make a bunch of copies of this RNA. So why does that matter? And so companies that are trying to develop RNA vaccines, what they're doing is they're taking the RNA that codes for these spike proteins on the coronavirus, which by themselves are not harmful. The harmful part is when you have the whole coronavirus, but not just the spike proteins. The spike proteins on their own are harmless. They can't infect cells. They're just going to elicit some kind of immune response. What happens next is they're going to take this RNA and they're going to put it into some kind of carrier. If we look at one of these red particles here and consider this to be a carrier, and specifically this would be an RNA carrier, so inside of it is going to be the RNA, the genetic payload that codes for this spike protein. What we do is we inject this RNA carrier into patients, and these carriers are generally things like vesicles, and they might have some kind of unique surface features that give them a particularly high affinity for certain cell types. And what happens next, we hope, is that a cell referred to as a dendritic cell, which I have right here in the bottom left, is going to perform something referred to as endocytosis or phagocytosis. And during this process, what happens is the dendritic cell, which has these dendrites, which are just basically all these little arm-like tentacle things, they're going to reach out and they're going to grab onto the RNA carriers. And once the RNA carriers are endocytosed into the dendritic cell, what happens next is inside of this dendritic cell, we want that RNA to get translated into protein, specifically that spike protein we were just talking about. And then once that spike protein has been translated, the next thing that's going to happen on that dendritic cell is it is a special type of immune cell referred to as an antigen presenting cell, or a professional antigen presenting cell. So it has things called MHC, major histocompatibility class I proteins that are meant to display other proteins on its surface. The spike proteins that were in that RNA are now going to start getting expressed on the surface of this dendritic cell. So what happens on this dendritic cell is now we've got a bunch of these spike proteins that are coming from the coronavirus that are being shown on the surface of it. And then the next thing that's going to happen is this dendritic cell is going to make its way to one of your secondary lymphoid organs inside of your body. And these are things like lymph nodes. And so inside of your lymph nodes, you have B cells, and B cells are going to feel the stuff on the major histocompatibility class I complex, and in this case it's going to be the spike protein. And if these B cells make something that has affinity for it, specifically if they're making antibodies that have affinity for this spike protein, they're going to turn into plasma cells, and these plasma cells are going to turn out a bunch of antibodies. And generally you'll see antibodies abbreviated as A(b). And so these antibodies are things that have a heavy chain as well as a light chain, and they've got this variable region right here and here. And these variable regions, which are referred to as the FAB, the fragment antibody binding regions, or antigen binding regions, are what's going to be binding onto your antigen. And in this case, the antigen, or AG, is going to be that spike protein from the coronavirus. And so these antibodies that are coming from your plasma cells are going to now have affinity for the SARS-CoV-2 virus, because on the surface of this virus are these spike proteins, and we now have antibodies. Your body's adaptive immune system now it has plasma cells that are making antibodies that have affinity for it. So what these antibodies are going to do is something referred to as neutralize the SARS-CoV-2 virus. And so what neutralizing antibodies do is neutralize antibodies. What they do is they're going to stick onto the outside of this virus, specifically those spike proteins. And because there are just so many antibodies that are stuck onto the outside of this virus, the spike proteins are no longer able to do their job of infecting host cells. And so because these antibodies are blocking those spike proteins from being able to grab onto that ACE2 receptor and help those fusion events happen with the virus in the host cell, these antibodies are now neutralizing the SARS-CoV-2 virus, and this is what's giving people immunity to it. And so in theory, this is how the RNA vaccines are working. The very important questions that we have yet to answer, and this is what's being done in our clinical trials, is whether or not these are safe and effective treatments or vaccines, and specifically some challenges that can be anticipated during this process are when we're designing these RNA carriers, we want things that are going to elicit an immune response, but not too strong of an immune response and not too weak of an immune response. So it's a Goldilocks dilemma where we are trying to figure out what is the best way to design an RNA carrier such that a dendritic cell or any other professional antigen presenting cell, like a macrophage, is going to want to eat it. That's a very, very big challenge. And how do you also make it stable enough so it doesn't just fall apart and it can make its way in an injection into a patient to actually get that vaccine or get that RNA to where we need it inside of these cells? The next step here is going to be what kind of RNA, what kind of sequence, what kind of structure do we want to give this RNA so that when the RNA is inside of these dendritic cells, how do we make sure that this RNA is actually getting translated into these proteins, because we need these proteins to get expressed onto the surface. So that is another very big challenge that we have to address here. And then a final question here, which we hopefully have already answered, which is what kind of protein specifically should we be targeting, because the coronavirus consists of many proteins, not just the spike proteins. Our hope is that because these spike proteins are on the surface, that they are a valid candidate for us to be designing RNA vaccines against. So if we can be more certain that this protein is exactly the one that we want to go after, if we can also be sure that the RNA that we have is able to successfully get translated inside of these professional APCs into proteins that are actually displayed on the MHC Class I complex of these dendritic cells, these immune cells, and if we're also able to make sure that we have RNA carriers that are stable and safe enough to be used in patients, then we might have a fighting chance. And so these are the clinical trials that are currently being performed, and I think this is really interesting stuff. Thank you all for watching. Please stay safe, wash your hands, take care, and I'll talk to you next time.